Hello there. Today in this lecture talk, let us understand the few question answers about the paper Molecular Biology. Today let us understand about Watson and Crick model of DNA. In the exam, you may get the question like explain Watson and Crick model of DNA. The Watson and Crick model refers to the groundbreaking model of the double helix structure of deoxyribonucleic acid proposed by James Watson and Francis Crick in the year 1953. This model revolutionized the field of biology and laid the foundation for our understanding of heredity and molecular genetics. With this note, let us understand in detail about this particular DNA. In the year 1869, biochemist F. Misher in the year 1869 for the first time discovered the DNA, okay? And he isolated an acidic substance rich in phosphorus, that is from the nucleus of pus cells and named as nucleon. Now let us focus on discovery of the double helix, okay? As I told, the James Watson and Francis Crick explored this particular DNA molecule in the year 1953 through X-ray diffraction, determined the geometry of DNA molecule. All right. Further, they received the Nobel Prize. They were awarded with the Nobel Prize in the year 1962. Watson and Crick, but not to Rosalind Franklin, who died of cancer at 37 from the X-ray data that provided the answers how exactly the DNA molecule look and their three-dimensional diagram, everything. Here you can see she's Rosalind Franklin who worked behind this particular DNA, and also X-ray data was given by this particular lady, but unfortunately she has not received any kind of award or any kind of credit. Okay. Further, let us understand about the Watson-Crick model of DNA here. In the year 1953, presented the double helix model of DNA, right? During this presentation, they also explained about the two primary source of information that is Chargaff rule. That is adenine always equal to the thymine and guanine always equal to the cytosine. That is purines, number of purines is always equal to the number of pyrimidines. A strange but possibly meaningless phenomenon, but this is applicable. Second one point, that is X-ray diffraction studies of Rosalind Franklin, Maurice, and H.F. Wilkinson. They provide the clear-cut, perfect, exact result of this particular DNA configuration. So basically, this is the DNA, what they have constructed, virtual DNA model done by Watson and Crick, who received the Nobel Prize in the year 1962. Okay, here you can see the Watson and Crick. Here they were so young. Here you can make it clearly. In the year 1962, they got the Nobel Prize in Physiology and Medicine, who only the James D. Watson and Francis H. Crick. Watson J.D. and F.H. Crick. Molecular structure of nucleic acids. They explained the molecular structure of nucleic acid, a structure for deoxynucleic acids that was published in the Nature 171 in the year 1953. He's James D. Watson, and here you can see the Francis H. Crick, Maurice H.F. Wilkinson, who worked behind that X-ray diffraction. And also, this is Rosalind and Franklin, but they all actually credited with the so much honors, but Rosalind Franklin, she did not get any kind of the honors or any kind of credits. Still, there is a question mark. What about Rosalind Franklin? Okay, so this is the structure of the DNA model, what explained by the Watson and Crick. This structure basically explains the key features, that is double helix, sugar phosphate backbone, nitrogenous bases like adenine, guanine, cytosine, and thymine, and also complementary base pairing, anti-parallel strands, major and minor groove, and dimensions. So all this was explained in this particular Watson and Crick model of DNA. The first and foremost is here double helix. Here, DNA consists of two long strands that wind around each other, forming a spiral shape, much like a twisted ladder. Okay, you can see here, much like a twisted ladder, you can see. That is about the double helix, 3' to 5' and 5' to 3'. Next one, sugar phosphate backbone. Okay, sugar phosphate backbone will be here, sugar phosphate backbone. The rails of the ladder are made of alternating deoxyribose sugar and phosphate groups linked by phosphodiester bonds. This forms the structural framework of the DNA molecule. Next, coming to the nitrogenous bases, which are hydrophilic, which are hydrophobic in nature, whereas sugar phosphate backbone, which are hydrophilic in nature, this but nitrogen bases, which are hydrophobic in nature. Here, always they pair each other. Pairs of nitrogenous bases that connect the two sugar phosphate backbone can be seen. That is, 4 nitrogenous bases, adenine, guanine, cytosine, thymine can be seen. And these nitrogenous bases are complementary to each other. We can see here complementary base pair. Thymine always base pairs with adenine with 2 hydrogen bonds. Cytosine always bases with the guanine with 3 hydrogen bonds. Okay, this specific pairing is crucial for DNA replication and the transmission of genetic information. And whatever here the 2 strands of DNA, they are anti-parallel strands. It means the 2 DNA strands run in opposite directions. One strand runs in the 5' to 3' direction, while the other runs in the 3' to 5' direction. All right. The 5' and 3' notations refers to the carbon atoms on the deoxyribose sugar. Apart from that, we can also see here major groove and the minor groove. Okay, this is the major groove. Deep groove is the major groove and the shallow groove is the minor groove. The twisting of the double helix creates 2 grooves. Okay, this is the major groove and here this side is the minor groove. A wide major groove and a narrower minor groove can be seen in this particular structure. And these grooves are very important for the binding of proteins that regulate gene expression and other DNA-related process. This major groove and the minor groove. When coming to the dimension, the double helix has a diameter of about 20 angstrom diameter. Okay, that is 2 nanometers. And each complete turn of the helix is about 34 angstrom. One helical turn for 34 angstrom, that is 3.4 nanometers long, and contains approximately 10 base pair per helical turn. Okay, for one helical turn, there will be presence of 10 base pairs. And the bases are stacked about 3.4 angstroms, that is 0.34 nanometers apart. Okay, you can see here 3.4 angstrom between each pair can be seen. Okay, this is all about your Watson and Crick DNA model. What you need to keep in your mind? The first you need to keep in your mind about the double helix. Okay, 2 long strands, sugar phosphate backbone. Always you can see here in this particular peripheral region, sugar phosphate backbone, which is hydrophilic in nature. Then in the center, you can see that in the central axis, you can see the presence of the nitrogenous bases, which are perpendicular to each other, which are hydrophobic in nature. And the most important one is chart graph tool, that is the base pairing can also be seen here. Then anti-parallel strands. One strand run in the 3' to 5', other strand run in 5' to 3' direction. The most important one, the major groove, that is the deep groove, and the shallow groove, the major groove and the minor groove, which play a very important role during the building of proteins, and also during the gene expression. And the next part you need to keep in mind about the dimensions. One each helical turn for 34 angstrom, where you could see the presence of 10 base pairs, and the distance, that is 3.4 angstrom between the pair, and the diameter of the double helix is about 20 angstrom diameter. All right. So this is all about this particular Watson-Crick model of DNA. This Watson-Crick model of DNA can also be known as B form of DNA. When we go for the different forms of DNA, we have A form of DNA, B form of DNA, Z form of DNA, and many more. Okay. Whereas here, this particular Watson-Crick model of DNA is actually the B form of DNA. Let us go through the explanation part of this particular Watson-Crick model of DNA. I've already explained in the previous slide only, but if you want, you can take the screenshot and just go through this one. So now let us understand this structure of DNA, what historiographic studies have revealed. Okay, and they have mentioned all this information. James D. Watson and Francis Crick proposed the three-dimensional model of DNA structure in the year 1953, and they awarded the Nobel Prize in the year 1960. This is not 1961, it's 1962. This model has the following features. The first one, according to this model, the molecule is formed of two strands, that is double helix, two long polynucleotide chains which are coil-like helix or spiral around the central axis, right? Second point, always the coiling will be right-handed direction around the central axis. It means this can also be known as the B form of DNA, right? So this B form of DNA is a right-handed direction around the central axis. Third point here, the framework of DNA molecule are formed of phosphoric acid molecule, deoxyribose pentose sugar, and molecule of nitrogenous bases. And the fourth point here, the hydrophobic bases, the central nitrogenous bases are hydrophobic in nature, adenine, guanine, cytosine, thymine, which are perpendicular to the axis, and a hydrophilic sugar phosphate backbone can be seen. They run along the axis facing the surrounding water. Okay, that's the fourth point. Then fifth point, in the helix, the two strands which are anti-parallel to each other. It means they run in opposite direction. That is, one strand runs in 5' to 3' direction. Other strands runs in 3' to 5' direction. It is anti-parallel nature. The sixth point, the nitrogen bases of two strands are paired through hydrogen bonds in the center. This is called base pairing. Base pairing in DNA molecule are highly specific. It means cytosine always pairs with the guanine, thymine always pairs with the adenine. Okay, if I'm going very quick, kindly take a screenshot and make a note of all these very, very important points. All right. Further, seventh point, always purine of one strand pairs with the pyrimidine of the other strand. That is, adenine always base pairs with thymine through two hydrogen bonds, and guanine pairs with cytosine through three hydrogen bonds. Thus, the two chains are complementary to each other, and this is known as complementary base pairing, which are highly specific in their nature. You can also recall here about the paragraph rule. The distance of coiling is about 20 angstrom. Okay. The helix takes one complete turn for every 34 angstrom. One complete turn will be for every 34 angstrom, where you could see the presence of 10 nitrogenous bases. 10 nitrogenous base pairing can be observed in one particular turn. And also you can see here major and the minor groove. Due to change in the geometry of the base pair, when the angle between the glycosidic bond is happening between the nitrogenous base and the deoxyribose sugar of the carbon 1 atom, right? The glycosidic bond is that is 120 degrees Celsius, 120 degree, it results in minor groove. When the angle between the glycosidic bond is 240 degree, it results in major groove. That is about the major and the minor groove. Okay, so this is all about the characteristic feature of the Watson-Crick DNA model, that is the B form of DNA model, or the characteristics. I hope you understood this one. The characteristics, you can divide that into what all? Please make a note. Double helix. About the two long strands are polynucleotide strands. Then you can talk about sugar phosphate backbone, then nitrogenous bases, complementary base pairing, anti-parallel strands, major and the minor groove, and about the dimensions. So all these you need to keep in your mind. Okay. I hope I made myself clear about this particular Watson-Crick model of DNA. I think there won't be any kind of doubt related to the Watson-Crick model of DNA. And by going through all these answers, whatever I've discussed with you, we can score very good marks. Prefer this one, that is more than enough. Here, the Watson-Crick model elegantly explained how DNA could carry genetic information through the sequence of its bases and how it could be replicated through the complementary base pairing. All right. Their work, along with crucial X-ray diffraction data, which was obtained from Rosalind Franklin and Maurice Wilkinson, earned Nobel Prize in Physiology or Medicine in the year 1962. There was a big question. All these three men got the Nobel Prize. At the very young age, she died of cancer while working with the exposing her body with the cancers, that X-rays, and she died of cancer. Nobody recognized this lady, Rosalind and Franklin. This unfortunately not acceptable. Okay, but it's okay. This is all about Watson-Crick model. This model remains the cornerstone of modern molecular biology. Without this particular DNA, we could not go for any kind of research work. 90% of the DNA is located in nucleus and the rest is distributed in cytosol and cellular organelles like mitochondria and chloroplast. In DNA molecule, adjacent nucleotides are joined in each chain by phosphodiester bond linkage or bridges between C5 of the deoxyribose, the mononucleotide, with the C3 of the deoxyribose base of the next nucleotide. Always one end of the nucleotide is 3' and the other nucleotide will be always the 5' free end. Okay. Between these C5, okay, always the 5' phosphate molecule and the 3' hydroxyl molecules are the two free ends for the formation of the polynucleotide chain. Then coming to the biological importance of the DNA, it's a genetic material in most organisms and serves to store transmission and expression of genetic information, right? This particular DNA with the sequence of nucleotides helps in protein synthesis. This particular DNA also explained about the Chagraph rule of base equivalence. Now let us go through in detail about the DNA functions. This in detail DNA function is not necessary, but still for your reference, please go through this one. Okay, if you mention few a function that is more than enough, but this is for your reference. Here, DNA's primary function is to store and transmit genetic information, right? You know this better. This particular DNA acting as the blueprint for building and maintaining organisms. It also plays a crucial role in protein synthesis, where it provides the instruction for the creation of protein, that is polypeptide chains, which are essential for various cellular functions. Our body basically more amount predominantly which is made up of protein than DNA molecule. Okay, DNA molecule which is large in the nucleus but enter body tissues, organs everywhere. We could see the presence of the proteins. Additionally, DNA is involved in replication, ensuring that genetic information is accurately copied and passed on to the new cells during the division. Let us study one by one the function, that is genetic information storage. DNA carries the instructions for an organism's development, growth, and reproduction. These instructions are encoded within the sequence of nucleotide base pairs. Okay, that's why we can say this particular DNA which carries the hereditary material which stores the genetic information. And this DNA also play a very important role in the protein synthesis. If there is no DNA, no protein synthesis. You know better about the central dogma. DNA to RNA, RNA to protein. That's why the prime role of takes that is about by the DNA molecule only in the biological system. Then here, the protein synthesis, DNA provides the code for synthesizing proteins, which are the workhorses of the cell, performing a wide range of functions like building tissues, transporting molecules, acting as enzymes, and many more. And this DNA also play a very important role in the replication, right? DNA can be replicated, meaning it can create an exact copy of itself. The same photocopy it can create. This is essential for cell division, ensuring that each daughter cell receives a complete copy of the genetic information. So you can keep in your mind the three important functions here: genetic information storage, protein synthesis, replication. If you can't go for explaining all this, just mention the heading. That is more than enough, sufficient. Okay. Next one is inheritance. DNA is passed down from parent to offspring, ensuring the transmission of genetic information across generations. Okay. So you can write about functions: genetic information storage, protein synthesis, replication, and the next one, inheritance, right? So this is all about the functions of the DNA molecule. I hope I made myself clear about the Watson-Crick model of DNA and about its functions. What does DNA do? DNA contains the instruction needed for an organism to develop, survive, and reproduce. Further, DNA is a biological molecule that contains the instructions of an organism needs to instructions to the organism what all needs to be functioned, develop, and for the reproduction. How does DNA function and how is DNA structured? The function of the DNA is to store all of the genetic information that an organism needs to develop, function, and reproduce, right? Whereas this particular DNA, which serves as the code for the protein synthesis, that is a polypeptide chain. What do you mean by a gene? A sequence of DNA nucleotides that code for one polypeptide. Okay, that is about the gene. Gene, you can also say functional unit of heredity, which stands for the one generation to other generation, which instruct the cell to synthesize a protein. You can give that particular definition also. And further, what is genome? Collection of the genes is known as the genome, or all the genes of one person is known as the genome, or collection of the genes is known as the genome. What do you mean by gene? Gene is a unit of heredity which starts from one generation to other generation, which instruct the cell to synthesize protein. So this is all about the Watson and Crick model of DNA. Let us go through very quickly what all we understood in this Watson-Crick model of DNA from the beginning. So went through about the Watson-Crick model of the DNA, right? Who explained this one? Who received the Nobel Prize in the Nobel Prize? Which year? And about the charter of full extent diffraction studies was explained by Rosalind Franklin, Maurice, and H.F. Wilkins. And this is the original DNA model, which was given by the Watson and Crick. Here you can see the most younger Watson and Crick original model, how they constructed over there. Okay, they received the Nobel Prize in the year 1962, and this is the basic general structure present characterization of the DNA model. This DNA model is also B form of DNA model, and further characteristics are given. Please take a screenshot and study. I explained also about the biological importance and about the DNA functions, and further about the few questions about the DNA molecule. All right. So this is all about Watson and Crick DNA model. Thank you everyone for watching this video. I hope I made this Watson-Crick model of DNA crystal clear. Thank you everyone for watching this video. Stay tuned with me for more videos.